Welcome to The Explainer. I am incredibly glad you're joining us today because we are unpacking a concept that honestly sounds like pure science fiction, but it is happening right now in laboratories all around the world. We are talking about the groundbreaking ability to directly edit the very DNA that makes us who we are. I mean, think about it. We're talking about a biological instruction manual with over 3 billion letters, and humanity just found the pen. Just imagine a world where doctors don't just, manage the symptoms of an illness, but actually erase genetic diseases before they even begin. Think about conditions like sickle cell anemia, cystic fibrosis, or even certain types of cancers being completely eliminated by simply rewriting a patient's biological code. What if we could literally rewrite the code of life?
Okay, let's dive into this. Here is our roadmap for today. We'll start by meeting the molecular scissors. Then, uncover how CRISPR edits DNA. From there, we'll see how it's rewriting human medicine, navigate the vital risks and ethical boundaries, and finally, look at the future of healthcare.
Section 1. Meet the molecular scissors.
So, let's look at the microscopic origins of this medical marvel, which actually start in a place you might not expect. This revolutionary tool was discovered in bacteria. Think of CRISPR as a microscopic filing cabinet. Bacteria have basically been fighting a turf war against viruses for billions of years. When they're attacked, they literally take a mugshot of the virus's DNA and file it away in their CRISPR system as a genetic memory. When that same virus attacks again, the bacteria use this CRISPR guide system along with an enzyme called Cas9, which acts precisely like a pair of molecular scissors to find and destroy the viral DNA.
Let's see how this builds from just a basic biological curiosity into a massive global phenomenon. Back in 1987, scientists first noticed these unusual repeating DNA sequences in bacteria, but they didn't really fully understand their purpose. Fast forward to the monumental breakthrough in 2012, when researchers Jennifer Doudna and Emmanuelle Charpentier proved that we could actually program this CRISPR-Cas9 system to cut specific targeted sequences of DNA. And wow, just 8 years later, in 2020, their work was awarded the Nobel Prize in Chemistry for transforming genetics forever.
Section 2. How CRISPR edits DNA.
Now it's time to understand the actual biological mechanics of how this microscopic tool functions in the laboratory. And this brilliantly illustrates the whole concept without getting bogged down in the dense biology. Just think of an everyday Word document on your computer. If your DNA is the entire document, your genes are the individual sentences making up that text. Mutations, the actual things that cause genetic diseases, well, those are simply typos in those sentences. CRISPR basically gives scientists a biological find and replace function to locate and correct those typos.
So how exactly does this biological find and replace work? It happens methodically in 3 distinct steps. Step 1 is target recognition. Scientists design a guide RNA which acts exactly like a GPS navigation system, seeking out the precise target sequence in the DNA. Step 2 is DNA cutting. Once the GPS finds the spot, those Cas9 scissors step in and quickly create a double-stranded break right in the DNA. And finally, step 3 is DNA repair, where the cell's natural mechanisms carefully kick in to modify or fix that broken gene.
Now, that third step, DNA repair, is absolutely crucial, because cells mainly use one of 2 different pathways to fix the break. The first is non-homologous end joining, or NHEJ. It's fast, but it's pretty prone to errors, and scientists actually use it intentionally when they want to knock out or disable a harmful mutated gene entirely. The second pathway is homology-directed repair, or HDR. This is a much more precise method where scientists provide a corrected DNA template, allowing the cell to perfectly correct the gene. And that right there is the holy grail for curing genetic diseases.
Section 3. Rewriting human medicine. These molecular scissors are actively moving out of the laboratory and into life-saving clinical treatments for real patients. Just look at the vast scale of these medical applications. The potential here is absolutely staggering. We're seeing research into cancer therapies by modifying T cells to better hunt tumors. We're fighting infectious diseases like HIV. And yes, we're even editing pig genes for safer organ transplants to solve global human organ shortages, literally making When Pigs Fly a little less fictional. But perhaps the most incredible victory so far is the successful application in treating sickle cell disease. Scientists are literally editing a patient's own bone marrow stem cells to increase healthy hemoglobin production, drastically reducing the disease's devastating effects.
Now you might be asking yourself, didn't we have gene editing tools before CRISPR? Well, yeah, we did, like zinc finger nucleases, but honestly, they were incredibly complicated. CRISPR completely democratized genetic engineering. It leaves older methods in the dust because it's exceptionally faster, dramatically cheaper, much more accurate, and incredibly versatile. It turned a process that used to take years into something that takes weeks, allowing laboratories all over the globe to jump in and participate in this genetic revolution.
Section 4. Risks and ethical boundaries.
Having this kind of unprecedented power over human biology obviously comes with severe limitations and intense dramatic tension. From a purely physical standpoint, scientists still face major hurdles to make this technology work perfectly in the human body. As outlined here, off-target effects are a huge concern. Like, what if the Cas9 scissors accidentally cut the wrong DNA sequence, potentially causing a brand new harmful mutation? There are also delivery problems, as getting these molecular tools safely into the correct cells using viral vectors is quite difficult. Plus, the human immune system might recognize the Cas9 protein as a foreign invader and mount a serious attack against it.
Beyond those physical hurdles, we must face a stark ethical contrast between two different types of editing. Somatic gene editing involves changing the cells in an individual patient's body, like curing a person's cancer. Those changes affect only that single person and are generally considered ethically acceptable. But in stark contrast, germline gene editing involves altering sperm, eggs, or embryos. Those genetic changes become inheritable, meaning they will be passed down to every future generation. This carries immense consequences and is heavily regulated or outright banned in many countries.
So, the crucial point is, what happens when we possess the ability to guide human evolution? Should we edit embryos to create so-called designer babies? While eliminating a painful inheritable disease seems incredibly noble, where exactly do we draw the line? What about editing for height, or intelligence, or eye color? The ethical weight of germline editing is a massive debate that humanity is navigating in real time right now.
Section 5. The future of healthcare.
Despite these massive responsibilities, the horizon of what is possible next is incredibly exciting. What's truly mind-blowing is that standard CRISPR-Cas9, that's really just the beginning. Scientists are already deploying next-generation tools. We now have base editing, which allows for single-letter DNA changes without making risky double-stranded cuts. There's prime editing. Think of it as upgrading from a blunt backspace key to a highly precise search-and-replace cursor. And we even have epigenetic editing, where scientists can turn the expression of certain genes up or down without actually changing the underlying DNA sequence at all.
Pulling all of this together, the ultimate takeaway is really clear. From personalized medicine and unprecedented cancer cures to future anti-aging research, CRISPR represents humanity's newfound ability to directly edit the very blueprint of life. It is the ultimate balancing act of wielding great scientific power while demanding great ethical responsibility.
We have covered a tremendous amount of ground today, from bacterial immunity systems all the way to the absolute cutting edge of human genetics. I want to conclude by leaving you with a lingering thought. Considering the incredible potential to eradicate immense medical suffering, weighed against the permanent, inheritable risks to our genetic code, would you support gene editing to cure inherited diseases? Think deeply about that. Thank you so much for joining me on this explainer, and as always, keep staying curious.